Hi, my name is Billy Taj. I'm with the Parkinson Lab at the Hospital for Sick Children here in Toronto, Canada. I'm here to present MetaPro, our in-house metatranscriptomic pipeline. As we know, metatranscriptomics has become the dominant technology in studying microbial communities. However, it generates huge amounts of data. Hundreds of millions of reads are common in a typical RNA sample. Processing times used to take weeks, if not months. This data also takes a number of non-trivial steps to filter, clean, and scan to gather any meaningful answers. Clearly, there needs to be a better way. Enter MetaPro, our massively parallel, fully automated metatranscriptomic analysis pipeline. The initial concept of MetaPro came from the metatranscriptomic workshop that we run most years as part of the Canadian Bioinformatics Workshop Series. We have access to a supercomputing cluster at SickKids, and in order to make efficient use of it, we redesigned the pipeline to be massively scalable based on the number of threads available on a server node that we have. MetaPro works like any other pipeline. First, we filter the incoming data for adapters, low-quality reads, hosts, and vector sequences, and we filter out reads that only contain mRNA. Then we combine the reads into contigs. At this point, we're left with contigs and reads that didn't quite match. When we run these two sets of data through our sequence aligners, which is a three-stage process using separate tools. After that, we run taxonomic classification and enzyme function annotation. Then after that, we compile the results into tables and statistical outputs for the user. Now, every step I've listed involves some sort of logic to transform the data file. Our secret sauce to making this process go faster is to subdivide the data at various sections and run them through each tool all at once. Wherever it's slowest, we split the incoming data into small chunks, then run those small chunks through the tool that we'll be using, and then recombine the data all at the end. We do this because the slowdowns are not uniformly distributed across the toolchain. So we won't split at every single step. We have painstakingly chosen, or rather chosen through experimentation, which steps and tools to apply this process to. Okay. Next is, now to compare the results between our pipeline, Human 3 and SAMSA 2, we sent a data set of 5 samples of kimchi with known lactic acid bacteria to see what each pipeline could pick up and in what quantities, displayed in this bar chart. The gold standard results were produced by running the samples through BWA with a reference database consisting of those known bacteria. MetaPro, in this case, picks up higher quantities of each known lactic acid bacteria, but compared to the gold standard, there's still clearly room for improvement. Now, the pie charts to the right represent the strength of the taxonomic classification tools of each pipeline. MetaPro was able to classify more of its data to species over the other pipelines. In the second trial, we sent 12 non-mouse gut samples with altered Shadler flora bacteria through each pipeline to see if they could be recovered. Here, both MetaPro and Human 3 detected a bit of Parabacteroides goldsteini. However, Parabacteroides goldsteini is closely related to the Parabacteroides ASF 519. Though Human 3 found no ASF 519, it does find a lot of goldsteini, and they are comparable. So we are saying that the blue is relative to this gold color. The results also speak for itself. MetaPro finds more ASF bacteria than the other pipelines. As for the taxonomic classification, MetaPro is still able to find, or rather classify, a high percentage of reads to a taxa over its competitors. And again, the gold standard was the raw sample sets run through BWA with the ASF bacteria in the database only. Finally, MetaPro also generates a good number of output tables and figures that can be integrated into other tools. This is a cytoscape figure, populated with the cytoscape table MetaPro creates from the enzyme annotation results. It imports neatly and without any effort. Now, we didn't need to reinvent any new processes here, but rather our aim was to modernize the workflow of metatranscriptomic analysis to the demands of today and the foreseeable future. We have made it possible to speed up the processing times by efficiently using multi-threaded computer architectures and made it freely available to anyone who wants to try it out. Please feel free to reach out to me at billy.taj@sickkids.ca if you want to learn more. Thank you for your time.